An acute panic attack presents with severe, immediate physical symptoms. Heart rate accelerates into tachycardia, the skin breaks out in heavy diaphoresis, breathing fragments into dyspnea, and the patient is overwhelmed by a profound sense of impending doom. The conventional allopathic model categorizes this experience strictly as a neurotransmitter imbalance confined within the central nervous system. Under this model, standard psychiatric management relies heavily on selective serotonin reuptake inhibitors and benzodiazepines to pharmacologically suppress the symptoms. The functional medicine paradigm approaches the pathology differently. In this framework, panic attacks are multi-system physiological alarms, not isolated brain disorders. To understand the genesis of these attacks, we have to map their system's biology. That requires tracing how gut permeability, genetic clearance bottlenecks, and hormonal shifts converge to trigger the brain. Suppressing the central nervous system with medication blunts the immediate sensation, but it leaves the underlying physiological dysregulations completely intact, maintaining the baseline threshold for the next attack. The anatomical trigger point for a panic attack sits within the brainstem in the locus coeruleus. This node is the brain's primary source of norepinephrine. These neurons are acutely chemosensitive, constantly monitoring the blood for slight changes in hydrogen ion and carbon dioxide concentrations. When a patient experiences systemic mitochondrial dysfunction or reactive hypoglycemia, cellular metabolism fails. To survive, the body is forced to rely on inefficient anaerobic glycolysis. This metabolic shift results in the immediate overproduction of lactic acid, which begins to accumulate in both the serum and the brain. Rising lactate mimics the chemical signature of tissue suffocation. When it reaches the locus coeruleus, it triggers a false suffocation alarm. This alarm rapidly strips away the inhibitory serotonergic brakes holding the system in check, forcing a massive, uncontrollable discharge of noradrenaline. The panic attack is the direct result of poor metabolic efficiency. Cellular energy deficits lower the neurological threshold required to spark this reaction, leaving the brainstem highly reactive to minor stressors. The genesis point for the systemic neuroinflammation driving these attacks is rarely in the brain itself. We have to shift the investigation to the gastrointestinal tract. The structural integrity of the intestinal epithelial layer depends on tight junctions, which are regulated primarily by the protein zonulin. Subjecting the body to severe physical and psychological trauma degrades these tight junctions. Crucially, this trauma simultaneously degrades the blood-brain barrier. A study observing military personnel during intensive survival, evasion, resistance, and escape training provides the clearest human model of this dual barrier breakdown under acute stress. This graph tracks the SARE study. A rapid spike in lipopolysaccharide binding protein indicates intestinal permeability. In parallel, a spike in S100B confirms a blood-brain barrier breach. Once breached, LPS endotoxins escape the gut lumen, enter the bloodstream, and physically penetrate neural tissue. Inside the brain, the endotoxins bind to TLR4 receptors on resident microglial cells, activating them to secrete highly neurotoxic cytokines like interleukin-6. A compromised gut lining acts as an open door. It physically invites systemic inflammatory mediators straight into the central nervous system, establishing a chronic stress loop that heightens anxiety. Inherited genetic architecture dictates how efficiently a patient can clear excitatory neurotransmitters from their system once a stressor passes. The COMT gene encodes an enzyme responsible for degrading catecholamines, which include dopamine, epinephrine, and norepinephrine. Its speed is determined by the Val158met polymorphism. Patients with a slow COMT variant operate with an enzymatic bottleneck. The pathway simply cannot process high volumes of excitatory chemicals quickly. During a stressful event, dopamine and norepinephrine accumulate. Rather than being cleared out, they back up and flood the system. This genetic accumulation traps the patient in a state of chronic sympathetic overdrive, destroying their ability to recover from stress. Environmental neurotoxins compound this issue. Heavy metals bypass the blood-brain barrier, embed in neural tissue, and inhibit mitochondrial ATP production, acting as a secondary bottleneck. Specific genetic polymorphisms and accumulated toxic burdens act as underlying hardware vulnerabilities. They leave the nervous system permanently primed for hyperarousal. Epidemiological data shows a distinct, severe spike in the prevalence and intensity of panic attacks occurring specifically in peripubertal females. This anomaly centers on allopregnanolone, a neurosteroid metabolite of the hormone progesterone. In an adult brain, allopregnanolone acts as a potent sedative. It binds to GABA-A receptors, enhancing inward chloride currents and hyperpolarizing the neuron to calm the system. But the neurodevelopmental shift of puberty triggers the expression of a very specialized subset of alpha-4 beta-delta GABA-A receptors in the hippocampus. When allopregnanolone binds to these specific immature receptors, a biological paradox occurs. The receptor reacts in reverse. Instead of enhancing chloride flow, it reduces tonic inhibition. A hormone that normally acts as a calming buffer instead violently triggers profound neuronal excitability and immediate panic. This occurs right as states of estrogen dominance during early puberty further saturate the already sluggish COMT clearance pathway. Due to this temporary receptor vulnerability, therapies that blindly attempt to boost progesterone in teenage girls often inadvertently provoke the exact panic attacks they are trying to cure. Synthesizing this functional architecture reveals a clear picture. An adolescent female experiencing severe panic is likely enduring a combination of the allopregnanolone paradox, a slow COMT mutation, and LPS-driven microglial inflammation. Functional medicine maps this precise biochemical individuality using advanced diagnostic assessments, deploying organic acids tests and comprehensive stool analysis to locate the exact physiological breakdowns. The data dictates the intervention. Resolving the panic might require stabilizing the glycemic curve with a specific nutritional protocol to prevent reactive adrenaline surges. It often involves targeted genomic bypass strategies, like administering bioavailable magnesium threonate to physically block hyperactive NMDA receptor pores in the brain. It requires daily heart rate variability biofeedback to mechanically strengthen vagal tone, reinstating the parasympathetic nervous system's top-down control over the locus ceruleus. By identifying these root causes, the clinical objective shifts away from lifelong pharmacological suppression of anxiety toward the active, precise restoration of systemic resilience.